Bio 121. We are moving on now in Chapter 7 to what we call cellular respiration. And cellular respiration, for the purposes of your understanding, by the time you finish this chapter, hopefully what you come away with is this idea that Mother Nature needs to collect and transfer energy in multiple forms, otherwise known as transforming it, such that at the end of the day, we can make potential energy in the shape of ATP. Right? It's ultimately what we're looking to do. And the basic equation that everybody's been teaching you since like 3rd grade on is you have some molecule, O6, plus a little bit of oxygen, which is why, of course, you are an oxygen breather, to give us some number of carbon dioxide molecules, some number of water molecules, and some number of ATP molecules. And, let's just balance this equation now. Let's see, 36, we need 6 of those. We need 6 of those, right, 2, 4. There you go, fantastic. All right, looks like it's right. Okay, between 30 and 32 molecules, depending upon the efficiency we're about to do. And so the real question you need to be asking yourself is, if this is what's floating around inside your bloodstream, we're going to call it glucose, and this is what you're inhaling as you're inhaling oxygen from the atmosphere, then somehow it has to travel to some location inside of your cells that will then complete the metabolism, okay, the anabolic and catabolic reactions to break this down in the presence of oxygen, such that you end up with this set of products. So if here are our reactants, here are our products, what's going to happen is we're going to have a series of steps which are going to make up glycolysis. Glycolysis. And as its name suggests, we're going to take this, we're going to cut it. And from glycolysis, we're then going to move into this thing that's called the citric acid cycle. I'll put CAC, or Krebs cycle. Okay? And then the products from both glycolysis and the Krebs cycle, or citric acid cycle, are then going to be transferred to this last thing here that's called the electron transport chain. And the electron transport chain, as its name suggests, is going to literally move electrons, store that energy such that we can then use it in the presence of oxygen, which is why you're breathing oxygen, such that we end up making that ATP. Now, that's a lot on one slide. And I understand, maybe you have a really good understanding of cellular respiration already, and you may not even need this type of introduction. But the idea, of course, is, remember, in our previous chapter, we have this thing that's called the sun. It sends out radiant energy. Whatever's on the surface of our planet, let's put it in blue now. Yeah, on the surface of our planet, primarily water, primarily phytoplankton are responsible for making the largest amount of oxygen on the surface of our planet. Every single cell on the face of this planet, with some exceptions, requires glucose as the primary energy source. And we take that glucose, and we started with 1,6 carbon structure. Okay, 1,6 carbon structure, that's what this stands for. And we convert it into 2 3 carbon structures. And once we've done that, we now can begin to, if oxygen is present, make the largest amount, concentration of ATP on the surface of this planet, inside of our cells, inside of the subcellular organelle, the mitochondria. And you might recall the story I told you about the mitochondria, how we picked it up years and years and years ago, okay? And now it's helping us make the resources to stay alive. So, having told you that, let's see what actually is going to happen next. Well, respiration has an overview. Cells extract energy from the food that you've ingested. As we work through metabolic pathways, notice there's a close link between the transfer of energy. Okay? So electrons have high energy levels, which means we can take advantage of that. And it's this incremental transfer of electrons that allows the cell to transfer food energy into ATP at those phosphate bonds. Remember, there's an alpha, a beta, and a gamma. It's the gamma bond we actually want to store the energy in for respiration. We can use it in many other ways elsewhere inside of the body. Collectively, we actually call these redox reactions. These are reduction-oxidation reactions where you couple. The idea is coupling small amounts of energy transferred in a directional manner such that you actually take a reducing agent plus an oxidizing agent. You make an oxidized product and you have a reduced product. Now this is the sort of place where students get lost rapidly. Molecules that gain electrons after the reaction they are reduced. And that means that they actually have more potential energy. Those that lose electrons, those are the ones that are oxidized. Think of it this way. They're literally burned. They're oxidized. They are set aflame. These are storing energy. And the whole purpose, of course, is that Mother Nature will transfer that energy as she moves along. Now, how do we do that? Molecules like nicotinamide, adenine dinucleotide, can occur in an oxidized state or a reduced state. So depending upon what its responsibility is, NAD+ is the oxidized state, the reduced state is NADH. You'll see in many textbooks that'll be NADH plus H+, where what means is we're moving protons around. NADH carries 2 electrons and 1 hydrogen proton more than NAD, and because of this, it's actually carrying extra energy. The intermediates, here's NAD to NADH. Electrons carry a very important responsibility, and what they're doing is they're shuttling electrons to electron transporters in where ATP is going to be produced. Now, there are other electron carriers, and what I want to make sure you see here is if here's NAD. NAD can also be found as FAD to FADH2, NADP, NADPH. Primarily, we're going to see these in distinct locations, so when you see them pop up, pay attention to where they are. Inside a living system, the hydrolysis of ATP releases energy into that system. Remember, we cleave this bond here, and we release that energy, and it drives reactions. The loss of a phosphate group is called dephosphorylation, and we're actually going to use this as a signaling system in the future. But right now, phosphorylation is the ability to add that, and phosphorylated molecules tend to be less stable and more likely to react, meaning they're unstable. And which means that when you find ATP with that terminal phosphate, it means it's really simple for it to let go of that phosphate. And if we ever got into the discussion of how muscle contraction occurs, how it is you literally move your body by creating small little machines, there's a series of machines that are enzymes that are responsible for hydrolyzing that phosphate, making it possible for you to move as rapidly as you move. Now, inside living systems, we do this inside the subcellular organelle, the mitochondria, which means ultimately we actually have to get from the cytosol of the cell, where glycolysis is going to occur, collect the intermediates, and then those intermediates will be sent to our mitochondria, where because the mitochondria is a dual membrane structure, where you have the outer mitochondrial membrane, inner mitochondrial membrane here in sort of green, and then the inner mitochondrial matrix on the inside, by partitioning chemical reactions, it allows Mother Nature to couple exergonic reactions in a process that's called chemiosmosis, so that you make ATP using this enzyme, ATP synthase. Most of the ATP inside of the body is made through chemiosmosis. It occurs not only in mitochondria, but also in chloroplasts and the plasma membrane of aerobic prokaryotes. So when somebody says it's an anaerobic prokaryote, you know it's not making ATP through chemiosmosis. Okay? So this is an important set of distinguishing characteristics. And if I'm you and I'm a student, I'm thinking to myself, hey, this is the sort of class where it's important to understand what these characteristics are. And you would be correct. So here's that equation. I've written it for you many times so far this semester. Here's that glucose. Here's oxygen. I forgot to put the 6 in front of the O2 there, right? Okay, and here's the six CO2s, 6 H2Os, and about 36 ATPs. Now, earlier I put 30 to 32, okay? And that's because, depending upon what we're actually thinking about and how we're doing it, the relative efficiency can get as high as 36 ATP molecules. It can get as low as 30 ATP molecules. But to get to that 30, if we were to have 100% efficiency, you'd have 100% efficiency in a series of 10 steps of glycolysis, a series of steps in the citric acid cycle, where the oxidation of pyruvate, which is a product of glycolysis, will allow materials to enter into the citric acid cycle. And then the products from both the citric acid cycle and glycolysis will then undergo oxidative phosphorylation inside of the mitochondria. And here's an important thing I want you to be thinking about, is now all of these are just words. But if you think back to what I'm trying to tell you, glycolysis occurs inside the cytosol of the cell. Oxidation of pyruvate is going to be a transition step to enter into the citric acid cycle. And the citric acid cycle occurs inside the mitochondria. That means oxidative phosphorylation is going to occur inside the mitochondria as well. And what you want to do is you want to partition these locations to understand the importance of oxygen in this entire story. So what is glycolysis? It's the first of metabolic pathway. There are 10 enzymatic reactions. If it was a biochemistry class, you would have to draw and memorize all of them. I've made students do that in the past, meaning biochemistry students, not biology students. For those of you who are thinking about medical school or something like that, you will go out of your way quietly to do it on your own. You cannot be thinking about your MCAT exams without having some real understanding of what each of these structures looks like. And of course, the inputs are going to be one glucose molecule. We'll have 2 NADH pluses, 2 ATPs, and 4 ADPs as a result of all of this. And you're thinking to yourself, what's going on here? Well, notice 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, somehow we actually have to go from this to this. 1, 2, 3. 1, 2, 3, 4, 5, 6. Remember, I did not lie to you when I said the purpose of glycolysis is to take one 6-carbon structure and convert it into 2 3-carbon structures. That's what we're looking for. They're called pyruvate. And by the time we've finished, we will have made 4 ATP molecules, but it cost us 2 ATP molecules. And we will have made 2 ADP molecules because we've used 4 ADP molecules. So, how? Well, here's a subset of all of the glycolysis steps. Okay, the first half of glycolysis involves 5 enzymes and uses 2 ATP molecules. Glucose becomes glucose 6-phosphate. The significance here is this. Once glucose is brought inside of a cell using hexokinase, it has to be phosphorylated so it doesn't move down its concentration gradient. Then we're going to take a isomerase. And remember what an isomerase is. It changes its shape. It's going to take glucose 6-phosphate and convert it into fructose 6-phosphate. Okay, so all it's done is swung some structures around. See if you can figure out which ones. Nudge, nudge, hint, hint, wink, wink. Then we use phosphofructokinase. And we've spent an ATP molecule here to make fructose 1,6-bisphosphate. So you can see that now this looks like a more symmetrical structure. And that fructose bisphosphate aldolase now is responsible for making dihydroxyacetone phosphate and or glyceraldehyde 3-phosphate. And you can see you can transfer between these two structures using triose phosphate isomerase. Now, here's what I'm not expecting you to do. I'm not expecting you to be able to walk me through this entire process. I am expecting you to know that glucose ultimately becomes glyceraldehyde 3-phosphate and or dihydroxyacetone phosphate. Okay, that's really important because now that we've actually made this molecule, G3P, we can now go through the second half where glyceraldehyde 3-phosphate in the presence of that dehydrogenase here can make 1,3-bisphosphoglycerate, and ultimately moving through the rest of these steps here. Notice what's happening. 1, 2, 3. 1, 2, 3. We still have 2 phosphate groups here. Ultimately, we get down to a structure that looks like it has 1, 2, 3 carbons, and there is your pyruvate. Okay? Now, without an ATP investment, it only produces 2 NADH and 4 ATP molecules. So that means Mother Nature has options, and she has multiple options here when she's doing all of this, and I need you to understand that. We teach you the canonical pathway because every single college biology class teaches the same canonical pathway, and should you go on further in this, you'll see where we can create, or where Mother Nature creates bifurcation points, so something else can happen. Ultimately, this is catabolism at its best. We've gone from a 6-carbon structure to a 3-carbon structure. And the catabolism inside anaerobic cells would mean that you don't need oxygen at this point in time. You don't. Notice we've not used oxygen at all. That's really important, which means should oxygen no longer be involved, this would then go into fermentation, okay, for glycolysis transferring into fermentation. So here are all the steps put together. You know, for those of you who are really ambitious, I recommend drawing all of this out. I will not be testing you on the individual enzymes as we're moving along. You should know glucose ultimately becomes pyruvate, though, and the intermediate to get there is glyceraldehyde 3-phosphate. Here's a great little video on YouTube to see how this actually works out. It's worth the time to actually take to watch it, but I'm not going to go through it here with you right now. We'll spend enough time talking about it in class. So what actually happens to that pyruvate then? So this is a pretty good place to pause, because what I want to do is I want to now think about what happens if oxygen is present or oxygen is not present. Okay?